The T-cell receptor is a complex of antigen-specific alpha and beta chains associated in the membrane with CD3 gamma, delta, epsilon, and zeta chains. Each of the CD3 chains has at least one copy of a signaling motif, the immunoreceptor tyrosine-based activation motif, or ITAM, in its cytoplasmic domain. T-cell activation requires the interaction of other molecules, such as CD45, whose cytoplasmic domain contains a tyrosine phosphatase enzyme, and the T-cell co-receptor, either CD4 or CD8. In this example, the co-receptor is CD4. The co-receptor molecules have the tyrosine kinase LCK bound to their cytoplasmic domains. LCK is attached to CD4 by a zinc ion, which bridges cysteine residues in each molecule. A phosphorylated tyrosine at one end of LCK is bound to an internal SH2 domain. This locks the kinase domain into an inactive conformation. In this example, the T-cell is a CD4 T-cell, and its antigen receptor recognizes an MHC class II molecule together with its bound peptide. When the T-cell receptor binds its specific MHC peptide complex, a number of events take place within the cell. CD45 dephosphorylates the terminal tyrosine of LCK and initiates its kinase activity. CD4 interacts with the MHC, bringing LCK into close proximity with the ITAMs of the T-cell receptor. Activated LCK then phosphorylates the ITAMs of the T-cell receptor. At this point, the tyrosine kinase ZAP70 is recruited to the phosphorylated ITAMs of the T-cell receptor and binds with specificity through its two SH2 domains. Now bound to the ITAMs of the T-cell receptor, ZAP70 is phosphorylated in turn by activated LCK. With ZAP70 now located in proximity to the inner cell membrane, it is able to phosphorylate LAT, a transmembrane protein with a large intracytoplasmic domain. When LAT is phosphorylated, the adaptor protein GADS binds to LAT and recruits SLP76, a cytoplasmic protein, to form a large scaffold. This scaffold will then be used in the subsequent activation of PLC gamma. While the scaffold can be generated by signaling through the T-cell receptor alone, subsequent events, including the activation of PLC gamma, require contributions of other co-stimulatory pathways involving CD28.